Hello, Mr. Allman here, and this is your Allmancast Introduction to Cells. In this unit, you're going to learn to appreciate how life is organized. From the organ systems of the blue whale, down to the microscopic structures of the E. coli bacteria, life compartmentalizes and specializes in ways that is so much more organized than the often chaotic environment that surrounds us. Humans have been studying the organization of life for as long as we know. But technology is something that needed to progress in order for us to appreciate what's going on inside of cells. And the world inside of a cell is something that helps you to truly appreciate how minute this organization really gets down to. Our tour of the cell is going to begin with some of the first people who studied them. Anton van Leeuwenhoek was a Dutch tradesman, not a trained scientist, but a glassmaker, and someone who was the first one to be able to develop really sophisticated lenses. And with those lenses, he was able to look at the microscopic world around him, and was the first person in recorded history who was able to accurately describe microbes from looking at things like pond water. A little bit after that, an Englishman named Robert Hooke actually coined the term cell. As he looked at the cork tree, he saw that there were these little compartments. And those compartments reminded him in shape to the honeycombs of a beehive. And so he used the word cells to describe how it is that this wood was divided. Later in the 1800s, a trio of German scientists were the ones that formalized our idea about the organization of living things down to the cellular level. They formalized their cell theory in three parts. First, all living things are made of cells. Plants, animals, protists, fungus, bacteria, all of them, whether large or small, can be broken down into their cellular parts. Secondly, the cell is the basic unit of life. One cell is capable of carrying out all of the necessary functions and processes in order to maintain and continue on life in future generations. Finally, all cells come from pre-existing cells. Most cells come about through the process of cell division, where mitosis and cytokinesis divide a cell to create a new cell that is a replica of the original. There are other processes that are going to allow for the creation of gametes, which are sperm or egg cells that will recombine to form new life. This idea was in contrast to a competing theory called spontaneous generation, which said that in certain circumstances, living things could come about from non-living material. So the advancement in technology is what allowed us to be able to see first and then understand exactly how it is that this life is organized. The unaided eye, or even simple lenses, weren't enough to be able to see most cells. Most cells exist in the 1 micrometer to 100 micrometer range, and the human eye isn't really capable of discriminating down to that level of individual cells. So the advent of light microscopes is what allowed us to be able to see microbes and individual cells really for the first time. Now, it's the addition in the 20th century of electron microscopes that allowed us then to be able to see the very smallest bacteria, to see viruses, and to see the more specific parts within cells that now we've come to know the function of. Now, life is divided up into cells for a very good reason. There are physical constraints on how big life can get. If living things are too small, then you don't have enough space to be able to fit stuff. The molecules that are needed to be able to carry out the building up of proteins, the things that are required to be able to digest food, the structures needed in order to be able to prompt the cell to divide. All of this stuff needs space. You need someplace to put it. Now, bigger, though, isn't necessarily better for a cell, because if something is too big, then you can't exchange matter, things like oxygen, carbon dioxide, nutrients, waste. Those things need to get into and out of the cell. And if the cell is too big, then that can't happen. And so being able to subdivide up a large living thing into smaller parts is a nice way to be able to increase that surface area so that exchange of matter, of nutrients and wastes, can happen much, much, much more efficiently. So surface area to volume ratio is a way to be able to quantify that relationship between how much space there is inside of the cell to fit that stuff and the surface area, which is going to be that amount of space that there is to exchange those things with the environment. So higher surface area to volume ratios are more advantageous to maintaining life because it allows for faster movement of those materials into and out of the cell. Now, cell types can be broken down into two main families. There's the prokaryotic cells. These are simpler cells that lack any membrane-bound parts called organelles. They tend to be smaller, typically less than 10 micrometers in size, but they're much more common. All bacteria are prokaryotic cells, and these are thought to be the first forms of life and the most plentiful and diverse forms of life. They exist everywhere, and they've been on Earth for almost as long as the Earth has been around. In this unit, we're going to spend much more time not on the prokaryotic cell and prokaryotic cell parts, but rather on the other member of the family, which are eukaryotic cells. Eukaryotic cells have structures that are bounded by membranes called organelles. So an organelle is just a small specialized part within the cell, and a membrane is going to be the barrier that separates the organelle from the rest of the environment surrounding it. They tend to be bigger, in the range from 10 to 100 micrometers on average, and there are two main types. The autotrophic or plant-like cells are going to be what makes up all plants and some of the plant-like protists, things like algae. In this case, some of the main differences are the presence of a cell wall, which is made of cellulose, which is going to be a carbohydrate that provides a lot of structure and protects against the loss of water for the plant. There's also going to be generally a large vacuole in the middle, which fills up with water. And this is going to be an important part that allows it to be able to maintain homeostasis, which is a important function of life where constant internal conditions are being maintained. By filling it up with water, it provides structural support for something like the stem of a plant and ensures that there's a constant supply of water available for photosynthesis. One of the other most important parts of plant cells and plant-like cells are the presence of chloroplasts, which are membrane-bound organelles, which have a set of flattened discs inside, and in those discs is where photosynthesis is occurring. They are also noted for their green color, and this is what ultimately makes plants green, are the chloroplasts within their cells. Now, heterotrophic or animal-like cells are going to be the main topic for this unit. Again, animal-like heterotrophic cells. We'll investigate in future videos a little bit more on some of the functions of these parts, but you should become familiar with being able to recognize them both in drawings such as the one here, as well as in microscope images, being able to name and identify the functions of the various parts within the cell. Now, animal-like cells are going to be made up of fungus, like the one that you see here, which do have a cell wall made up of a different structure called peptidoglycan, as well as animal cells, which make up animals and animal-like protists, including things like the protozoan, the paramecium. Now, in this case, it's easy to be able to see the structure, and you can see one of the modifications that's used to be able to increase that surface area. This is the lining of the uterus, and the lining of the uterus needs a large surface area, and that surface area is accomplished by being able to fold the outer membrane. And that folding of the membrane allows for more diffusion or movement to occur from the environment outside of the cell into the cell itself. So a couple of points I want to bring up in closing this introduction. So the first is why it is that membrane-bound organelles are used. Well, organelles are going to help cells to be able to specialize. They isolate areas, allowing for certain conditions to be held within them. For example, an acidic pH inside a digestive organelle in which molecules are going to be broken down and digested. They also provide surface area for reactions to occur, in particular cell respiration, which is the metabolic process by which food molecules are broken down to release energy, as well as photosynthesis, which is the process by which plants and plant-like protists are going to be able to create food using energy from the sun. These reactions need a lot of enzymes, which are located on membranes, and so that space is needed to be able to accomplish those reactions. This gives complexity and allows for specialization. And that specialization is necessary in order to maintain multicellular life, because not all cells within a multicellular being are capable of carrying out all of the life processes, but rather certain cells do certain jobs. And so the types of organelles present within a cell say a lot about what it is that the job of that particular cell is going to be. So, for example, if you have a heart muscle cell needs to be able to process energy at an extremely fast rate, because those heart muscle cells are constantly at work throughout the course of their lifetime. And so they're going to have a large amount of organelles that are going to provide energy in order for them to continue doing that job. So having a lot of compartments gives a lot of options, allows for different types of cells and for cells to perform different functions. Finally, as we look at the life of the cell, going back to the cell theory, and the cell is the basic unit of life. Cells need to do all of this stuff and more, in many cases, to be able to stay alive. They need to be able to gain nutrients, so they need some way to be able to bring food in, whether that's food being made within the cell through photosynthesis, or food that needs to diffuse or actively be brought in to be digested as a source of energy. Things need to be metabolized. So food molecules need to be broken down, and new special molecules, things like proteins, carbohydrates, fats, and nucleic acids, they need to be made. And so the synthesis of those are summarized in the whole set of chemical reactions happening within the cell, which is known as metabolism. Cells need to respond to stimuli as conditions change in the environment, or as signals are passed from neighboring cells in multicellular organisms. Cells need some way to be able to sense and receive that stimuli or that communication, and then process it and respond to it accordingly. So the movement of a paramecium could be an example of this. As the paramecium has cilia that sense the presence of food, they can use those cilia and move them to propel them towards that food source to be able to gain the nutrients that they will then metabolize so that they can have the energy needed to survive. All cells will grow and develop. These are two different concepts. Growth is about getting bigger, whereas development is about changing and becoming more complex. So cells, when they first form, through cell division or otherwise, those cells need to physically get larger, and by taking in food and adding on to themselves. But cells also may need to develop to make new organelles, new parts, become more specialized over time. Finally, cells need to reproduce. They need to be able to copy their own genetic information and pass that on to new generations of cells. And so all of those systems exist and work within these tiny, tiny little microscopic compartments, the complexity of which we're only beginning to scratch the surface of. Thank you for listening, and please let me know if you have any questions.